is known as the micropylar region, and which exists on seed coat is the micropyle just above the hilum. Seed structure. A typical seed consists of the following parts: seed coat, embryo. The seed coat, it is the protective coat. The seed coat is the protective coat made of the two layers. Testa, is the outer layer, is also called as testa, and the tegma is the inner layer. The present of the envelope to protect the embryo and endosperm from the dissection. The mechanical injury effect of the environmental fluctuations and damage due to the insects and microorganisms. It also helps the dispersal of the seed. Seed coat also they can use for the dispersal of the seed. Embryo, it is the rudimentary plant present in axis form with the one tip known as the plumule, responsible to the form shoot portion, and the other tip known as the radical forms. The root, the portion of embryonic axis extended above the cotyledon is known as the epicotyl, and below the is also called cotyledon is the hypocotyl. Below the hypocotyl, above the epicotyl. Okay, this is the structure of the seed pod. See, this one is the this is the structure of the seed pod. See, this one is the diagram of cotyledons and the epicotyledon, the above the portion. Epicotyl is the extent embryonic axis, and next one is below the cotyledon is also called as the hypocotyledon. Okay, epicotyledon, epicotyl, and hypocotyl. What are the store food storage of the food? Endosperm, it is a thick and massive structure made of the elongated cells containing abundant starch. On the base of the presence or absence of endosperm sheets are the categorized into two groups. One is the albuminous, is also called as the endosperm present. Albuminous cells is also called as the endosperm is present. Exalbuminous, endosperm absent. Okay, the cotyledon is the extension of the embryo. On the base of number of cotyledons, the crop species are the divided into two groups. One is the monocotyledons. Monocotyledons mean only they are containing only one cotyledon is present. Dicotyledons is also containing the two cotyledons. Some of the gymnosperms have the 2 to 18 cotyledons. Monocot is the scutellum is the situated in the between the endosperm. The embryo is the single cotyledon. Albuminous and exalbuminous. Albuminous is the monocot, weight, rice, and other cereals. Some of the examples, different categories of the seeds, the albuminous and exalbuminous. Monocot is the wheat, rice, and other cereals. Exalbuminous onion. Dicot, the castor is the albuminous, the feed. Okay. Availability of the endosperm is present, also called is the albuminous present. Monocots, wheat, rice, and other cereals. Exalbuminous, exalbuminous means the endosperm is the absent. The monocot, the example is the onion. And dicot, albuminous is the castor. Castor is the castor. Next one, exalbuminous, the P and other pulses is also an example. It's very important competitive examination point of view. Okay, now what is the fruit structure is known as the seed. The single-seeded fruits are usually considered as a seed. Caryopsis, single-seeded fruit with the fused fruit and cereal, wheat, sorghum, pearl millets also. Akin, the single-seeded fruit where the pericarp can be removed from the mature seed. Example is the sunflower, safflower. Akins, the fruit, the actually caryopsis, the single-seeded fruit is mainly that the containing the of the Poaceae family members, caryopsis. Akin also, the single seed fruit is the pericarp can be the removed from the mature seed. The example is the sunflower. Next, hydrocarp, a dry fruit which is the separated into two or more units the at maturity. Example is the coriander and carrot. It belongs to a Poaceae family members. Nut fruit with the thick and hard shell like the fruit was the groundnut. These are the nut structure of the crop seeds. The monocot seeds of the crop from the family of the Gramineaceae. These are the caryopsis. Endosperm is the seeds is made of the non-living cells bearing the alveolar layer, outer layer. A very small embryo made of the shield shaped the single cotyledons known as the scutellum and a short axis lie in a groove at one end of the endosperm. The epithelium layer separates the embryo and endosperm cotyledons form the protective sheath known as the polioepitel and the plumule and the poliorized radicle. Monocot is the crop from the family Graminae. Beat is the hump at the dorsal, the trace at the ventral side. Embryonic end of the seed is known as the polar end, whereas the other end is known as the brush end. Rice is the caryopsis, tightly closed to the lemma and palea. The endosperm of the rice is of the two types, mainly high breaking breakage during the milling and Eperius, the less breakage and non-sticky. The sorghum pericarp consists of and the epicarp, monocarp and endocarp. The hilum turns the black at the physiological maturity. The endosperm consists of a layer, the alveolar cell, and on the outer corneous endosperm surrounding and the central flori are the stachey endosperm. Mass seed is the conical is in shape with the broad and the round upper end of the sharply pointed lower and embedded in the mouth. White and the oval area contains a ridge-like structure, which is the indicates the position of embryo. The yellow part of the grains is endosperm. Others onion is the actually onion is the non-albuminous. This is the monocotyledons, exalbuminous cells. Seed. Embryo is the spirally twisted and cylindrical in shape. Most of the part of the embryo is made of the cotyledon. Dichosis of the crops from the family Leguminosae. These are the exalbuminous dicotyledon seeds. The embryo is made of the embryonic axis with the two fleshy cotyledons. The seed is the round to oval embedded by the protective seed coat, the scare of the hilum and the micropyle. Radical portion of the embryonic axis projects the behind the cotyledons. The other end of the embryonic axis is the plumule present between the both cotyledons. In some species, what like the outgrowth is the present the below hilum is called as the strophiophilia, and others onion, and dicot seeds crops from the family Leguminaceae, or the akins, the sunflower, safflower, niger. These are the albuminous dicot single-seeded. The fruits covered with the fruit wall having some ridges. Usually, a very thin layer of the endosperm is present in between the outer covering of the seed and the cotyledons. The cotyledons are the flat with the clear nerves. The embryonic axis is the short and the radical is present in extended form at the one side of the it castor. It is a large seed covered with the hard seed coat. At one end of the seed caruncle is present as whitish outgrowth of integument. The hilum is the present at the base and partially hidden into the caruncle, whereas the micropyle is located in between the caruncle and the hilum. Testa as the white and brown spots, the removal of the testa, while the blank, the flattered endosperm covered with the thin stigma layer becomes visibly. Embryo is embedded in the endosperm. The two cotyledons are the large and flat with the clear wings. The plumule bears a number of mi- minute leaves near the apex and with the remains in between the two cotyledons. Chestnut, the flattish ovate white into light brown small seed with the dark base, pennate marginal line and an equally pale central line in the one face. Seed of the exalbuminous is the straight embryo, linseed, the plant to the elliptical, the ovate brown in the glossy seed. Mustard, the plant is the epithelial, the spirate to the irregularity globulus, dark reddish brown, brown black seed, the stand visibly absent. Accuse lactica sativa, seed with the both endosperm and the cotyledons as stored as food. Bit, beta vulgaris seed with the endosperm, cotyledons, and the nucleus tissues of the store the food. Coffee, the nucleus of the ovary after the fertilization becomes the perisperm and stores the food reserves. Coconut, it is unusually endosperm with the pot of liquid and the cellular cucurbits. The seed is the flattened and the ovate with the smooth surface seed coat thick. Two cotyledons comprise the seed. The seed dormancy. Seed dormancy can be defined as the state or the coordination which seeds are the prevented from the germinating even under the favorable environmental conditions. Types of the dormancy. Exogenous dormancy. Next one is the endogenous dormancy. Exogenous dormancy means the physical, mechanical, and chemical. For the physical, the seed coats are the impermeable to water. Seeds are the at low moisture level. Legumes are the example. Okay, legumes, the example of the physical, the exogenous dormancy. The embryo is the generally inactive. The cause of physical dormancy is the structure of the outer cell layer, which becomes impermeable to water. Macrophage cells, glycerages, other cell layer are hardened in the endocard. Three reasons the seed spore becomes the impermeable up to water. The seed pods they develop during the last stages of the seed development. A study with the Coronilla varia seeds showed that the depth of the puncture to the seed coat increases so did the permeability of the seed coat to water. Mechanical dormancy. Seed coats are the too hard to allow the embryo to expand during the germination. The causes include the structure of the seed coats and the remaining fruit occurs in folding. In nature, the coats are the softened by the environmental agents such as the acid, nuts, microorganisms in warm, moist in environment, forest fires, to overcome the horticulture scarcity, the stand up for hot water, acid, warm moisture environment. The fire is the nature push. Once scarified, the water can get in, have to be the careful because the seed is also more susceptible to damage by the microorganisms. But the next one is the chemical, endogenous and exogenous. Exogenous is the physical, mechanical, and chemical. The presence of the chemical inhibitors in the outer coverage of the mini fruits and the seeds. This occurs in fleshy fruits, hulls, and the capsules of many dry fruits. Examples are the apples, citrus, grapes, and desert plants. Very often, see this kind of the dormancy disappears with the dry storage. This kind of the dormancy may also be present in the other tissues surrounding the embryo, such as the endosperm. In addition, oxygen may be restricted by the mucilage layers of particles spinach, some of the oscillations. In nature, overcome by the heavy rains, which can leach the inhibitors in tropics, deserts, some soils absorb the toxins such as the ammonia. To overcome the horticulture leach, the running water change water daily for several days. The chilling of the 2 days existing embryo remain on street is the harmonious, especially GF. All of the types of the exogenous physical, mechanical, and chemical. Endogenous dormancy, morphological dormancy. Morphological dormancy, embryo is not fully developed at the time of the ripening. Needed additional embryo growth after the seed is the separated from the plant. The terms of the how much space is the embryo takes up in the seed, and several herbaceous flowers seeds such as the ranunculus, poppy, woody species such as the holly, the tropical plants such as the date plants. When the combined with the other kinds of dormancy such as the hot seed force, rudimentary about the pro-embryo stage, maybe the inhibitors present, ranunculaceae, pepaveraceae, and arelicaceae, the linear, in the torpid stage, takes up about the 1 by 2 of the seed cavity, maybe inhibitors the present, umbelliferae, ericaceae, primulaceae, and differential the orchids. To overcome artificially alternate warm and the cool temperatures, also hormones such as the GA3 exposure to cool temperatures, high temperature for the full development of the embryo. So with the date palms, it often takes several years to the germinate. Takes a long time to reach the full embryo. Development often high temperatures, the 100 degrees Fahrenheit will accelerate this. Next, how to break down the seed dormancy methods. The physical method, the scarification. And treatments may be the physical or chemical, but the weakness or the softness of the seed coat is known as the scarification. Acids, the mechanical, the physical treatment, hot water treatment, the biological treatment also, acid scarification. By using the concentration sulfuric acid, 100 ml of the 1 kg of seed, in 2 to 3 minutes, the treatment dormancy can be overcome in the above group of the seed. The duration of treatment will vary and it depends on the type and the nature of the seed coat. The tree crops of the example tree crops 1 to 3 hours. The rose seeds treats with the partially with the acid and then the given with warm stratification. Mechanical scarification. Seeds are the rubbed on the sandpaper with the help of the mechanical scarifier or by the puncturing on seed coat with the help of the needle to the enhance. Increasing the moisture absorption by the seeds. Example, bitter gourd for the sand scarification and sand and the seed 2:1 ratio should be followed. The rub against the hard surface of seed for a 5 to 10 minutes. Biological scarification. It is done by the digestive systems of the animals with the help of the guts in the dodo birds. Hot water treatment. It is the effective in case of the leguminous tree crop seeds. The seeds should be the soaked in the boiled water for 1 to 5 minutes. Some crops like the Bengal gram and the Bengal gram and the groundnut hot water treatment for the more than the 1 minute the found injuries of the seed. Hot water treatment. Stratification. When seed dormancy due to the embryo factors, seeds can be subjected to the stratification treatment. Cold stratification. Incubate the seed at low temperature of 5 to 0 to 5 degrees cover over the moisture substream for the 2 to 3 days to several months of the it depends on the nature of the seed and the kind of the dormant seed, cherry and oil palm seeds. Warm stratification, some seeds are the required the temperature 40 to 50 degrees for the few days. Example, the paddy in case of the oil palm, it requires the temperature 40 to 50 degrees for the 2 months, the breaking of the dormancy. Care should be the taken during the treatment and the moisture content of the seed should be not more than the 15 percentage. Bleaching of the metabolites inhibitors. The seeds can be soaked in the water for 3 days. But once in the 12 hours, fresh water should be changed to avoid the fermentation of seeds. Can be soaked in the running water for a day to leach of inhibitors. For example, coriander, sunflower, leaching metabolic metabolites inhibitors. Temperature treatment. Low temperature treatment plants which is the grow in temperate and the cooler climate require the period of the chilling for the breakage of the dormancy. For example, apple seed dormancy can be released the low temperature treatment by the storing the seeds at the 5 degrees centigrade. High temperature treatment. Normally, high temperature treatments are the exhibited by the early flowering winter annulus, bluebell, Acanthidius, Nanoscorpia, that the seeds are the shed in early summer and not the germinate until the have been the exposed to the air during the high summer. Alternate temperature treatment. Most of the plant species which grow in temperate and the cool temperate regions required alternate temperate for the breakage of dormancy. Bulrush, typha. Alternate temperate treatments. Most of the plant species which grow on the temperate and the cool temperate regions require to the alternate temperate for the breakage of dormancy. Bush. Fire treatment. Many shrubs and the trees are the subtropical and semi-arid regions have the extremely hard seeds in which the seed coat is the very improves of water dormancy, such as seeds is clearly coated and imposed in the may be broken exposure to extreme heat such as the fire. Light and the phytochrome. For example, inactive form of the phytochrome, red light is 680 nanometer, then active the PFR phytochrome, the non-dormant. Red light promotes the germination and the far right light is the active the germination. Okay, these are the seed structure and dormancy. Next one is the seed storage. Next topic is the seed storage. You need to seed storage is the prevention of seed with the initial quality until the needed of the for planting. Gene banks conserve the genetic resources. The most fundamental activity in the gene bank is to treat a new sample in a way that will be prolong its the viability. It's a long possible while the ensuring its quality. The samples are the monitored to ensure that they are not losing viability. The seed gene banks standards was first set by the International Board of the Plant Genetic Resources, the International Plant Genetic Resources Institute, IPGRA. Now, the Biodiversity International on the basis of the report from the PAO. Expert consultation group which meet the Rome, Italy from the 26th May 1990. The most widely used the technique for conserving the plant genetic resources in seed banking. Seed bank. Seed gene banks are set up with the objective to minimize the loss of viability and genetic integrity of seed and material during the storage and regeneration. Seed gene banks. Seed gene banks also, there are the three types of the seed bank in the storage. The short term, mid term, next one is the long term storages. Short term storages, these are the generally maintained at the temperature of 15 to 20 degrees centigrade and relatively humidity of 30 to 40%. The breeders, the materials and germplasm materials in the processing for the long term can be stored in the short term storage modules. Medium terms of the storage, the temperature of 0 to 10 degrees and relative humidity is the 25 to 30% and seed moisture is the 6 to 8% and adequate for the medium term storage module. It can maintain the seed longevity for the period of 25 to 35 years. The storage modules are meant to maintain the active collection, medium term conservation and regeneration, multiplication and distribution to bonafide the users and the evolution or other important activities. The accession size the compresses of the large a quantity as possible to the avoid frequent regeneration. Relatively humidity controlled through the dehumidifier since relative humidity is controlled, the seeds of the medium term storages can be kept in the paper bag, cloth bag, or any other moisture previous containers. The viability of the seeds in the monitored in the interval of 5 years. Long term storages. The storage condition in the long term refers to a temperature 10 degrees to 20 degrees minus 20 degrees and no control of the relative humidity. Only the base of the collections are the maintained in these conditions and can be maintained the seed anywhere from the 50 100 years. The base collection and it can be maintained the seed anywhere from the 50 to 100 years. The base collection with the minimum of 200 seeds for the self-pollinated crops and 4,000 seeds of the cross-pollinated crops are the generally performed for the storage in case for some of the wild species. 500 seeds must be stored. The seeds are the dried to the moisture content of 3 to 7 percentage and the seeds are the stored in the hermetically sealing of the container. Standards for the seed gene bank. Seed drying, the seed should be the dried prior to place in the storage bags, the containers. Sun drying, shade drying, natural air drying, hot air drying, and is the desiccants in the sealed containers are the generally used. Lower the seed moisture for the prolonging the longevity of the seed using the Embrify or the drying the seed at 10 to 20 degrees, 25 degrees and 10 to 15% of the humidity. Seed cleaning. The seed to be the stored should be the free from the weights, pests, and diseases, crop specific knowledge and weights. Pests and diseases associated with the seeds are the preferred. Storage containers. The storage containers are the different for the three types of the storage purposes. Generally, paper bags or plastic bags are used in the short-term storage. Plastic containers and plastic pouches are used in the medium-term storage. Where in the long-term storage, the modules, aluminum containers, and the multi-layered pouches are polythene are used. Monitoring of the seed viability. The minimum acceptability limit of the generation, the 65 to 85% of the gene banks. It will be assessed through the germination test. A minimum of 100 to 20 seeds are the 1, 1 kg used for the germination of the start of the seed storage. The period of the viability monitoring is 10 years for the base collection and the 5 years for the active collection. Regeneration. Regeneration should be the undertaken when the viability falls below the 85% of the seeds, 100 plants or more seeds of the generation. To avoid the large losses of the enlist, seeds used to for the generation should be the genetically as close as possible to the original sample. Information management, plant genetic resources and database is very important at national and international, regional and the global level of the conservation of rapidly disappearing the genetic stocks for the possible future using the crop improvement programs. The following databases are the important for the management of the seed and gene bank. Germplasm passport database, germplasm characterization database, germplasm evolution database, gene bank conservation databases, germplasm exchanges, maintenance of the modules, safety and security of module. Seed packing. The seed packing involves the placing or counted or the weighed sample of the seeds in the container, which is the hermetically sealed for the subsequent storages. Seeds are the packed to the prevent absorption of the water from the atmosphere after the drying heat accessing the separate and the avoid mixturing of them. The prevent contamination from the insects and diseases. Time and seed packing. The best time packing seeds are the immediately after moisture content has been determined to be the within the required limits for the safe storage. Dry seeds will reabsorb the moisture from the more humid ambient air. So the seeds should be the packed with the waterproof containers and hermetically sealed without the delay of the following removal from the drying room or the cabinet. Of the processing and the treating of the completed seeds of the pack into the containers of the specified new weight. Packing or bagging of the essentially the last operation seeds of the and in bulk flow. The packing of the consists of the following operation: filling of seed bags to an exact weight, placing leaflet in the seed bags at the regarding the improved the cultivation practices, attaching labels, certification tags on the seed bags in the seal of the bags, storage and shipment of the seed bags. Normally, seed can be the packed into the two methods. One is the oxygen filtered bags and drums with the oxygen supplied from the cylinder. Among the above two methods, oxygen implanted bags with the sufficient water is the considered good due to the advantages like reduced damage, the seeds and the easy handling feature the seeds of the patches bags. Therefore, when it mass mortality occurs at the The effective only lesser number of seats. The whole lot, the drum may affect the whole lot seats are that are the packed in the drum. The seeds can be transported in the carrier either by planted bags or in the oxygen supplied drums. The distances to be the main base of the following: external temperature, number of seeds in the bag, size of the seeds, metabolic wastes that is the are the released by the seeds, nature of the seeds, the possibility for the repacking the routine of the storages, storage at the ambient temperature and the humidity. Dry storage with the control the moisture content but not the temperatures. Dry storage with the control the both moisture content and the temperatures. Dry storage for the long-term gene conservation. Moist storage without the control the moisture content of the temperatures. Most moist cold storage with the control of the temperature and cryopreservation, also called the cryogenic storages. Seeds are the place the liquid nitrogen at the 190 degrees centigrade. Seeds are the actually placed with the gaseous phase, liquid nitrogen, minus 150 degrees centigrade for the easy having and safety metabolic reactions come to the rituals stand still at the temperature of the liquid nitrogen. The cells will remain the unlated state until the tissues move from the liquid nitrogen and deforested. Therefore, the little determined the physical activity takes place in these temperatures with the lungs, the storage like this, not the particle for the commercial seed storage, but it is useful to the storage of the availability of plasma. Storage containers, moisture purvies. It still storage the facility for the controlling temperature and the humidity. Then the permeable containers can be safely used for the orthodox seeds, the several areas, provided that the pets can be excluded. The cotton bags, cardboard, rubber-boarded moisture impervious of the drying the orthodox seeds to correct the moisture content. Seeds can be replaced in the seed, the moisture proof containers. This avoid the extensive dehumidification requirements. Long-term storage is the most effective in the moisture proof containers. Are combined with the controlled low temperatures provided by the refrigeration. This method has and the added advantages of the exclusion of the oxygen is not suitable for the reclaimed seeds or the Evo 700 Tango polythene bags. Moisture resistant. They include the polythene or the other plastic film. Moisture resistant. That include the polythene and other plastic films, the lignifier. That are resistant to the passage of the moisture, but over the a long period of the time, this will be the slow passage of the water, vapor tending at equilibrate the relative humidity inside and outside of the container. Polythene is not the suitable for the long-term storage and the orthodox seeds for the genetic conservation because there is no absolute control the over moisture uptake by the seeds. It is the very much suitable for the short or medium term storage as give the excellent results. Equipment used for the packing seeds, the bagger waiter, the manual weighing, the automatic seeds, mm, bag swing machine, labeling. These are the responsible for the making of the for the R labeling. Contents of the marks are the label. There shall be specified and the very every mark or label. The particulars are specified by the central government under the clause B in the section 6 of the Act. It is a correct statement of the content in terms of the weight, and expressed in the metric system. Data of the testing. If the seed in container has been treated, a statement indicating that the seed has been treated. The commonly accepted the chemical or abbreviated chemicals name of the application, the substance, and if the substance of the chemical used for the treatment and the present with the seed in the formal and human beings or other the vertebrate animals, the portion of the statement such as the do not use for the food, feed, or the oil purposes. Manner and of making or labeling of the container and the class C of the section N and the class B in section 70. This is the certified labeling. And this is the labeling, certified seed tax labeling, certified seed labeling. Mainly seeds, the show should be the for the future use. The seeds are the uniquely equipped, the survive as the viable regenerative organism. Next is the physical and biochemical changes and the during the seed storages. During the seed storage and seed moisture content and the temperature are the responsible for seed deterioration. Many physiological and the biochemical changes occur in the seed during the seed aging. Retroactive changes enhance when the seed exposure the external changes occur the seed during the seed aging. Retraction enhances when the seed exposure the external challenge increases the and annual losses due to the retroation can be such as the 25% of the harvested crop. It is one of the basic reasons for the low productivity sheller